Hello, today I'll be discussing my research presentation topic, optimization of transferrin receptor or CD71 monomer expression in mantle cell lymphoma. Starting out with some background on the topic, iron is a very essential element for many physiological processes of the human body, and particularly the cellular metabolism and proliferation. As far as iron uptake, this is achieved through transferrin receptor, CD71, when transferrin-bound iron binds to the transferrin receptor CD71 and is then endocytosed into the cell, and then the vesicle can be acidified and iron released intracellularly for intracellular processes such as cell cycle regulation, transcription, cell growth, and cell survival. CD71 is known to be present on many types of cells, specifically in the bone marrow, red blood cells, placenta, among others. CD71 is also highly expressed in rapidly dividing cells. These can include abnormal uncontrolled malignant cell proliferation and normal physiological renewal. Dysregulation of iron hemostasis occurs in cancer cells, allowing for the rate-limiting enzyme in DNA synthesis to have a continuous supply of iron with upregulated CD71 expression. Numerous studies in the past have demonstrated that cancer cells have higher CD71 levels compared to normal cells, and that higher CD71 expression is correlated with more advanced cancers. It is unclear, however, to what extent CD71 expression occurs in lymphoma. In our previous in vitro study, it was observed that varying levels of CD71 expression occurred in monomer and homodimer forms for lymphoma cell lines, as CD71 is two monomers bound together by disulfide bonds. The goal of the study is to optimize current methods of detecting CD71 expression in proliferating lymphoma cell lines to further delineate the expression that occurs in lymphoma. Following biochemical principles, we believe that changing the denaturation, time duration, and temperature would allow for possible further linearization and free disulfide bonds and hydrostatic interactions from the monomers to be broken up and to be read more accurately. So this led to the first question: what is the optimum denaturing temperature to achieve monomeric CD71 in mantle cell lymphoma, or JCO1? Second question was, what is the optimum denaturing time to achieve monomeric CD71 in mantle cell lymphoma, JCO1? For our methods of CD71 denaturation, we used JCO1 cell lysates that were denatured using 5x fluorescent master mix coming from a pre-packaged kit, and also used 400 millimolars of DTT to break down the disulfide bonds and 10x sample buffer. Incubation of denatured CD71 from JCO1 cell lysate was performed by thermal cycler at variable set temperature and time duration. For immunoassay-based CD71 quantification, expression levels of CD71 were measured by simple Western analysis, a capillary electrophoresis-based immunoassay. Using previously optimized assay lysate and antibody concentrations, we assembled the anti-CD71 assay with capillary wells that were loaded with 0.3 micrograms per microliter JCO1 cell lysate and 10 microliters anti-CD71 at 1 to 100 dilution. The level of CD71 in each capillary was quantified with area under peak around 104 kilodaltons and 180 kilodaltons, representative of monomer and dimer forms respectively. As you can see on the right-hand side of the slide is an electropherogram displaying this. Results for denaturing temperature of 75 degrees Celsius can be seen in the charts below. Of note, the dimer area was the lowest at 75 degrees Celsius for 30 minutes, and the largest monomer area was seen at 75 degrees Celsius for 50 minutes. Signal-to-noise ratio was greater than 50 for all results, telling us that there was no interference with this run. The graph on the left represents the data from the prior slide of CD71 monomer versus dimer area expression. The graph on the right demonstrates CD71 monomer over dimer ratio. For the CD71 monomer-dimer ratio, we see that 75 degrees Celsius for 30 minutes had the optimal lowest dimer and the highest monomer. Results for denaturing temperature of 70 degrees Celsius can be seen in the two tables below, with notable dimer area expression lowest at 70 degrees Celsius for 35 minutes and largest monomer area expression at 70 degrees Celsius for 60 minutes. To the left of the slide, we see the CD71 monomer versus dimer area, and to the right of the slide, we see the CD71 monomer versus dimer ratio bar graph. With the ratio, it is noted that at 70 degrees Celsius for 35 minutes, there was the largest monomer and lowest dimer formation. Results for denaturing temperature of 65 degrees Celsius can be seen in the tables below, with notable dimer area at its lowest expression at 65 degrees Celsius and 33 minutes, and monomer area expression greatest at 65 degrees Celsius for 60 minutes. To the left of the slide, we can see the CD71 monomer versus dimer area for denaturing temperature of 65 degrees Celsius, represented previously in the table on the last slide. On the right side of the slide, CD71 monomer dimer ratio for 65 degrees Celsius can be seen, with noted monomer to dimer ratio being greatest at about 65 degrees Celsius for 30 minutes and 65 degrees Celsius for 35 minutes. Results for denaturing temperature of 65, 70, and 75 degrees Celsius can be seen in the two tables below, with noted lowest dimer area expression at 75 degrees Celsius for 15 minutes and monomer area greatest expression at 65 degrees Celsius for 50 minutes. To the left of the slide, we see the CD71 monomer versus dimer area that was previously represented in the last slide. To the right of the slide, we see the CD71 monomer dimer ratio for the 65, 70, and 75 degrees Celsius denaturing temperatures, with noted ratio greatest at 75 degrees Celsius for 50 minutes, with lowest dimer expression and highest monomer expression. Discussing the results of the research, when comparing the denaturing temperatures, it appeared that with decreasing temperature, monomer expression decreased as well, and an increasing denaturing time was associated with increasing monomer expression to a certain point, but when reaching 60 minutes, monomer expression decreased. This is likely due to the bonds from hydrostatic forces and disulfide bonds reforming. And based on the denaturing results, it was concluded that the optimum temperature and time of denaturation for JACO1 or mantle cell lymphoma was 75 degrees Celsius for 50 minutes. Discussing the limitations of this research, the assay measured total CD71, dimer on cell surface, and monomer in cytoplasm. Also, measurement on primary cell lines may closely mimic the disease, but also it may not actually completely represent it. The assay is not sensitive to any mutation on CD71 protein, which could be added into the overall expression of CD71. In conclusion, the elevated levels of CD71 expression, particularly monomer form, on malignant cells, such as mantle cell lymphoma or JACO1, with its extracellular accessibility, ability to internalize, and central role in cancer cell pathology, make this receptor an attractive target for future antibody-mediated therapy. For example, programmed death ligand 1 or programmed cell death protein PD1 have both had similar research done on it before to quantify transmembrane protein expression with increasing precision. Today, we now have anti-cancer targeted agents for these specific receptors. Thank you for your time and consideration.